yang bermanfaat buat kita semua, dan terima kasih buat Dea, terima kasih buat juga Ade dan juga Om Begin, Bang Begin ya, yang senantiasa selalu mengawal scores gitu ya sampai ke yang 14. Jadi ke depan kita mungkin akan lebih banyak lagi ya share pengalaman, dan teman-teman yang ada di sini gitu ya, mudah-mudahan bisa mengikuti dengan baik. Itu saja, jadi mudah-mudahan skor kali ini bisa berjalan dengan baik. And Mas Lionel, thank you so much for your time. Thank you. Ya, silakan Mbak Dea dilanjut.
Thank you, Dr. Dia, thank you Pak Syamsul, and thank you Trias for the invitation. So hello, my name is Lionel. I work as a research fellow at the Tropical Marine Science Institute at the National University of Singapore, or NUS. I was from the Reef Ecology lab, so that was last year before my thesis submission, but we still all work together anyway. So today I'm going to present to you what we are doing in Singapore the past 2 or 3 decades with respect to coral reef restoration. I understand from Tria's massive paper recently that you all have a even much longer history of restoration in Indonesia. So for Singapore, it really started around the late 90s. And I'll give you a rough overview about what we have done so far. But then to start a bit about me, apart from the time that I spent in the office, doing paperwork like the rest of you all, I also spend a fair bit of time underwater monitoring stuff. I do reef surveys, find out how the Singapore reefs are doing, if they're not doing well or if they're bleaching. My team sounds out to the rest of the researchers in Singapore, and we try to do something about it. I'm also interested in marine biodiversity in really urbanized marine environments like recreational marinas, because contrary to what we think, you might think that the marinas are full of pollution, but surprisingly, there's quite a lot of biodiversity there. And of course, for the purposes of today's talk, I'm also interested in restoration, how to best design strategies to ensure that the coral reefs in Singapore thrive and become more resilient. So apart from staying in a field, getting wet, I also spend a fair bit of time at the St. John's Island National Marine Laboratory. It is, of course, on St. John's Island, which is the south of mainland Singapore. It is Singapore's only offshore marine research station, and there are a number of natural marine habitats around it, which gives us a lot of research potential. We have a lot of skilled researchers. It's open, the facility is open to all researchers. So perhaps after this COVID pandemic is over, you guys can come over and take a look. And we have a number of facilities that might be of interest to you. The first would, of course, be aquaria tanks. We have them indoors and outdoors to run whatever experiments you want. We also have controlled environment aquaria where you can tweak parameters like temperature, salinity, acidity to run the mesocosm experiments. We also have a research vessel named after a particular genus of coral. We had a naming competition many years back and the coral people won. That's why it's called Galaxia. So we also have a few state-of-the-art research laboratories, outreach programs to educate the public, and also a lot of skilled people to facilitate your research. So to know more, just check out this website over here at SGI NML. Okay, so I will go into the topic of today. So today I understand I'm supposed to talk about coral reef restoration, but it's fortunate that you guys are a school of coral reef restoration, so I don't have to go too much in detail about the importance of restoration because you guys know as well as me, or even better. But we just know that reefs they occupy a very small extent, at 0.1% of the world's surface area, but they are very important, right? They support the world by giving a lot of ecosystem goods and services. They contribute a lot to tourism, for example, a lot of materials provided, a lot of seafood. The estimated value is worth hundreds of billions annually. But as we all know, there's a lot of unsustainable resource use, a lot of destructive fishing practices, for example. And add to that, we have ocean sprawl, which refers to the proliferation of urban structures in the coastal and marine environment. So when we add all these impacts together, there's a very high percentage of coral reefs that are lost or threatened. And there are also global studies looking at this, and evidently over the course of time, you see that many major groups of coral reef animals have declined. For example, fishes, in response to coral reef loss, the response is generally negative across different trophic groups for fishes. So all this happens because there are these multiple stresses acting together, or it could be due to coastal protection or legislation or enforcement being a bit not very effective, or it could be that natural recovery is too slow. Whatever it is, it's all these stresses that act together that cause the impact. And we know very well that if you do nothing, the reefs will really decline all the way from coral-dominated areas to algae-dominated areas. And so this is why we look to coral reef restoration to try to bring the degraded ecosystem back to what it once was, or at least put back the structure and functions that have been reduced. And so recently there have been a number of reviews done, and this paper shows that most of the restoration work done, they aim to create a self-sustaining, functioning reef ecosystem again. And they do this by a variety of methods. Very commonly, we have artificial reefs, we have corals grown in nurseries, which are then transplanted out to places that need them. Increasingly, you see a lot more restoration work done. And of course, our region has a very high percentage of the work done. Of course, Indonesia dominates. You have so many reefs and so many projects ongoing. And from the 1990s, you start seeing a lot more work. Most of it is experimental, which makes sense because different sites require different strategies, and people just need to test out what works for each of their sites. So we go to Singapore, which is at the southern tip of Peninsular Malaysia. For Singapore, since the 1950s and 60s, there's been a lot of rapid economic development and population growth. Our waters are very busy. If you look out to the Singapore Straits from the south, you can see a lot of vessels, a lot of cargo, oil rigs being built and ferried around. So very busy port waters. Our land area has also increased by a lot. Since 1950s or 60s, it's increased by more than 20%. So if you see this dark brown area, this is the land area in 1953. And as time goes by, you see the area increasing. So we often joke that drawing the Singapore map is very easy, getting easier and easier because the lines are very straight now. So by 2030, it's projected that we will have a land area of about 760 square kilometers. And all this is to support our population growth and economic development. And if you've been to Singapore along the beaches, you would see urban structures such as these used for the purposes of recreation or economic development. So what does all this mean for reefs? Our top problem would be the high sedimentation and turbidity. Of course, sedimentation and turbidity resulting from coastal development, sedimentation falls on the corals and smothers them. The turbidity reduces the light penetration, and so photosynthesis is affected. So what happens is the reef belt becomes more and more narrow. So according to our big boss, Prof. Chow Lok Ming, when he was a kid, I think in the 1940s or 50s, he says that the visibility was very far, like 10 meters, and the reefs went down to more than 10 meters deep. But right now, our reefs mostly terminate around 6 meters. The reef crest is about 2 meters. And average visibility is also about 1 to 2 meters. So you can see that our reef belt is really shrunken and very narrow now. The next problem would be that our reef framework has gotten quite unstable. There's a lot of loose pieces of rubble lying around. And coral larvae that settle on these pieces of rubble, you can imagine that they are at risk of dying, right? When the currents move them, they might get killed. So when you add these together, it results in more than 60% decline in reef area since the 1950s. And we do expect more declines because according to our Ministry of National Development, this is the land use plan in 2030. And you can see that in the southern islands, where most of our reefs are, there are projected reclamation plans Yeah. So we do foresee some of these things happening, and this is why we are looking to restoration to try and rehabilitate the environment. At this point, some might ask, is restoration still relevant? Do we still continue doing it? We say yes, because despite the impacts, we still have many species in Singapore. Every now and then we have new records coming up. For reef fishes, we have recorded over 100 species. For sponges, over 200 species. And for hard corals, historically, more than 250 species have been recorded in our little island. So species richness still is quite high. There have been 3 major bleaching events since 1998 and a number of minor bleaching events. But after every bleaching event, the reefs still manage to recover somehow. And every year around this time, we have our annual mass spawning event. And so based on all these reasons, we say that active restoration is a viable intervention for Singapore to help to rehabilitate Singapore's reefs. So how do we do it? We first focus on 2 main problems in Singapore. The first would, of course, be your unstable substrate. The second problem is your high sedimentation. There are 2 main approaches. The first is a more physical approach, where we try and improve the quality of the substrate. We basically make the substrate more suitable for coral larvae to settle, establish, and grow. And the second is a more biological way, where we directly put down corals to increase coral cover. So I'm going to be talking about these 2 approaches broadly. Okay. So for the first approach, where we are looking at artificial reefs, we are looking here at reintroducing structural complexity and increasing substrate availability. So it's of course been used for fishes, where you create a lot of microhabitats for fishes. But for corals, what happens is you provide a stable substrate for them, and they are then able to establish and grow. And I know that you have many kinds of artificial reefs in Indonesia, even more than what are pictured here probably. And these are all made of really heavy and stable materials, right? Like concrete, metal, all these, so that they rest on the seabed very well. But for Singapore, we can't really put large concrete blocks down because our reefs are very narrow. If you use a barge and put these concrete blocks down, you end up destroying more things. So what we did in the early 2000s was to design this kind of artificial reefs, which we call reef enhancement units, or REUs. So they resemble flower pots that are about the height of your knee or your waist, depending on how tall you are. And they have inclined surfaces, so sediment doesn't accumulate, it just falls off. There are holes at the side which allow water to pass through. And importantly, they are actually made of fiberglass and not concrete. So because it's fiberglass, it's very light, you just need 1 or 2 divers to carry them to particular sites and then anchor them down with metal sticks at the areas that need them. So 4 months after they were deployed, they noticed that there were early colonizers on these structures. You get a lot of algae, various types of algae actually. And between 6 months to a year, you start seeing coral recruits. And these are the different types of coral recruits that you see close to a 2-year mark. However, this project ended in the mid-2000s and there was no funding after that. And so we did not continue monitoring. But fortunately, in the mid-2010s, we managed to secure some funding to revisit these structures because we're wondering how are they doing. Every now and then we swim past them, but we never really quantified the long-term effects of putting these artificial reefs down. So in 2014, we went to check them out again, but unfortunately, only slightly more than half of the RUs remain. A lot of them have been dislodged, possibly due to inadequate anchoring or because of strong waves, which really tells us that we really need to assess the structural integrity of these artificial reefs very regularly, or for any restoration effort at all. So you need to really go and check every now and then to make sure that things are all right. But for those that remain, we did notice very rich abiotic communities on them, a lot of anemones, soft corals, and hard corals. And when we compared the communities on these units with those 10 years ago, we find that the communities are different. And interestingly, the hard corals in 2014 are now about 10%. So roughly, we needed about 10 years for hard corals to inch up to around 10%, which tells people that biodiversity development takes time. And this is something that we try to tell our stakeholders when they talk to us, when they ask us for advice on biodiversity enhancement, that there's no quick fix. All these things require a lot of time, a lot of effort, a lot of monitoring. So when we also compared the differences in the RU plots across the different islands, we noted that there were differences. So the RUs in some islands, they supported more hard corals. In other islands, they support more turf and mixed algae. Others, they supported a mix of coralline algae and hard corals, which tells us that the location that you put the artificial reefs is actually very important because different sites, they support slightly different assemblages. And we think that this could be due to differences in water quality and hydrodynamics. Certain sites, such as Raffles, they tend to be larval sinks, which could explain why the coral diversity here is so high and the coral cover is so high. So when you looked at ecosystem functions, yes, these RUs do provide quite important ecosystem functions. In 2014, we noted that from 10 genera of corals, they were sexually mature. They were ready to give off eggs, which means that they are actually contributing genetic material to seed the surrounding reefs. They also provided food and habitat for more than 119 reef organisms, from damselfishes that were laying their eggs on the internal walls of the RUs, to cowries and urchins that were grazing on the walls, lobsters that were using the RUs for shelter, and of course, curious cuttlefishes checking out the insides of the RUs, possibly for food or to a place to lay their eggs. So you can see that there are really many reef organisms that attempt to use these RUs for and to make a living. So over time, we just started, every time we swam by, we just take a picture of a few RUs just to see how they looked. In 2002, of course, this is what they look like, largely nothing. In 2014, when we did the monitoring, this is what some of them looked like. 2018 to 2020. So if you remember a few years ago, there was this hashtag 10-year challenge thing, right? Where people will post pictures of themselves 10 years ago and now. This year happens to be our 20-year challenge for the RUs, and this is what they look like now. So if you're interested to find out more, you can check out this paper over here or email me for it. So the next approach is has got to do with increasing coral cover. This is where we create more coral material to repopulate degraded areas. How? By growing them in nurseries and transplanting the corals out to sites that need them. We know that coral nurseries get our inspiration from plant nurseries. So like plant saplings, you grow them in little pots. Once they grow big enough, you either sell them or you eat them, right? So for coral nurseries, the idea is to provide a stable shelter environment so that the corals can grow. And in Singapore's context, it's really important that we reduce the impacts of sedimentation. So we have two types of nurseries in Singapore. The first would be the ex situ nurseries. This refers to the aquarium tanks at St. John's Island National Marine Lab that I mentioned. For this, we are able to pump in filtered seawater, and we can do a variety of experiments, including sexual propagation. So what we do is we bring up the adult colonies before they spawn, collect the spawn, let them fertilize, and then we get the recruits and we monitor them. So one of the interesting things that we observed was that if you feed the corals, they tend to grow faster and they survive longer. I mean, a bit like humans, right? We eat more, we grow bigger. Not so sure about the surviving longer, but yeah, for corals, so far it's okay. So this benefit actually translates to when you transplant them. So the recruits that have been fed actually fed better in terms of growth and survivorship after they have been transplanted. So feeding is a useful strategy for us in terms of restoration in Singapore. We also do a lot of asexual coral propagation. About 10 years ago, when we were using these tanks, we were rearing the corals on racks, about 1 or 2 tiers in these kind of tanks. But recently, we started exploring the use of toy bricks, where we just have to cut out a piece of fragment, epoxy it to the toy brick, and string it up in our tanks and monitor their growth. So what this does is it's a modular system. So the larger the coral grows, we just have to add more bricks onto it to accommodate it. And it's also very space efficient. We are able to use the entire water column in the tank. So like Singapore, everything is space limited. Our tanks at St. John's, we also lack space. That's why we have to do this strategy. So our aim is to actually populate our 5,000 liter tanks with as many corals as possible so that we can support other researchers' work, contribute them to conservation, and possibly get a lot of basic data where we can then pump into research, try and make the aquarium trade more sustainable. So those were ex situ nurseries. Now we talk about in situ nurseries, which are in the sea. So like I mentioned just now, sedimentation is a problem in Singapore. So we made nursery tables, which look like this. They're elevated from the substrate, so sediment doesn't get to the corals. And there's a plastic mesh on the table, so sediment just falls through. So I also mentioned the corals opportunity, which are loose fragments lying on the reef, which might otherwise die. So rather than let them die, we decided to collect them and use them to stock our nurseries. So we found that this was actually a very useful way, in the sense that we don't waste the corals. We noted that the corals were growing quite fast. Different species, of course, grew differently, from 2 times increase in diameter to up to 12 times increase for other species. And how do we know that the nurseries work? We tested out the transplants and compared those that have been nursery reared to those that were directly transplanted. And yes, the transplants that were nursery reared actually fared much better in terms of growth and survival than transplants that were directly placed at the site. So we also realized that nurseries, apart from propagating corals, they are also able to provide other ecosystem functions. So the corals there provide substrate for other organisms to lay eggs. There are juvenile fishes gathering around there, and even larger organisms hiding under the nursery tables. And so we also noted that the different growth forms of corals that we grow in the nursery actually support different organisms. For example, the branching corals naturally support a lot more crabs than the massive corals. And all this tells us that the nurseries provide functions beyond just growing corals. They really are a hub by themselves. They can contribute a lot more ecosystem functions. So what do we do with the corals that have been grown up and they are of a suitable size? Of course, we transplant them using marine epoxy. This is inert epoxy that cures in half an hour, and over time the coral will grow over the epoxy. So these are a lot of growth experiments. Through these experiments, we are able to understand how fast certain species of corals grow. For example, the Porites over here, it's about a tennis ball size in 2009. In 2014, it became the size of a deflated rugby ball. Others, like this Echinopora, took 6 to 8 months just to carpet the entire piece of bare rock. So this one grows very fast. We also noted that they did also provide a lot of ecosystem functions like habitats for worms, food for parrotfishes. So for Singapore's context, of course, sediment is an issue. In one of our recent studies, we tried to investigate how to improve post-transplantation growth and survival. And for this, we tried it out at a site where sedimentation was a problem. It's very thick, a few cms thick. For this, we tried to transplant corals near to each other and corals further apart. And we found that at about 3-4 months into the transplantation, those that were transplanted close together, they started joining together. And about 15 to 16 months into the study, you start seeing that actually those that were not transplanted together, there was higher mortality than those that were transplanted together. So this tells us that for such an environment that's very high sedimented, it might be worth trying such a strategy where you group them together so they are stronger together. And we also tried to calculate some of the costs and realized that if you join the transplants together, at least for this Platygyra, you do have quite a bit of savings, about half compared to if you transplant them separately. So you can find more details in this paper. So this is another video of another of our projects. We were trying to populate this site. It was largely a barren kind of seawall area. Not much corals then, but and then we shifted corals from a site that was going to be destroyed by coastal development. So the authorities, they discussed with us and we shifted some corals over there. So this is at about the 3-year or 4-year mark. You can see that the corals have grown over the epoxy. You cannot see the white epoxy anymore, and they're growing on the substrate. And we transplanted many different species of corals. All of them were from the site that was meant to be developed, and they're doing well now. So for this same project, 3 years into the project, we were proud parents. We noticed that our transplants were actually sexually mature, and they were actually spawning in 2019. So we were quite excited to document that. So at this point, we were then beginning to wonder, can we create new reefs? Well, let's recap what we have learned so far. We know that our coral propagation techniques are effective. So for example, this is a picture of a capra not on a reef, but actually on a nursery table. So these started out from fragments about 10 cm, and now they have carpeted an entire nursery table about 1 by 1 meter. For this other species, Echinopora, it also took about 3 years to grow very big and over the nursery table, also about 1 by 1 meter. So yes, our propagation techniques for Singapore are effective. We also note that corals can colonize artificial substrates in Singapore. For this, we make observations of corals growing on the seawalls in Singapore. We noted that corals are actually growing on seawalls in the east, in the south, and all the way in the west, which means that these seawalls do function as very stable substrates for corals. Apart from that, we also noticed that there are a lot of corals growing in the recreational marinas, on the sides of birding pontoons and on the seawalls. So combining these observations together, we decide to, in our largest endeavor to date, these are our 10-meter tall artificial reefs, which we designed along with the government bodies. These are anchored at our Sisters Island Marine Park. So the surfaces are very rough. These are to encourage different kinds of reef organisms to settle, especially corals. There are platforms here which are able to receive corals from relocation projects and which we are monitoring right now. And you see all these pipes, they are meant as habitat for different sizes of fishes. So currently there are about 8 structures and we are currently monitoring them. And this is what a regular day is like monitoring them at the artificial reefs at Sisters Island Marine Park. Visibility isn't the best, but thank goodness for technology that we're able to color correct, so the corals look quite okay. And in general, they are doing quite well. They're all growing quite, they are growing larger. It's very obvious to us. And recently, we had 2 clips produced by the mainstream media. If you're interested, you can just Google Channel News Asia and this show called Wild Urban Designs or Me Watch. But if not, you can approach me and I can pass you the URLs. So there are snippets of the artificial reefs in these videos. So I mentioned also that we have a number of recreational marinas. You would think that these marinas, the water quality isn't that fantastic, right? But we also decided to try. So we installed some floating structures on the pontoons and put down some of the coral fragments. And we noted that they are actually growing quite well. And those that have grown a certain size, we transplant them to the seawalls in other parts of the marina. And yes, the corals still grow. So this strategy also works for marinas. So if you remember, we have a lot of seawalls in Singapore, more than 60% are artificial now. A lot of them are either sloping seawalls and vertical seawalls. So how do we make use of what we know to try and enhance marine biodiversity even more? First, you have to consider the limitations of what we can do in this type of seawalls. So when the tide recedes, there's actually a very limited space that is covered by the water, right? So when light hits, there's only a certain zone that gets illuminated enough such that corals can grow into a community. Same for these sloping ones, even though there's a slightly larger surface area than the vertical ones. So what if we actually design seawalls that favor marine biodiversity? What I mean is we actually can introduce the idea of tidal pools, where we have big pools which retain water even after the tide goes down. So desiccation, high temperatures, all these is less of a problem compared to if the seawalls were featureless. So that's sort of our dream, and we are optimistic. The other thing to get people more interested to nature will be to bring them closer to nature. And for this, we envision having sort of coral gardens attached beside pontoons, where people don't have to get wet. They can just walk along the pontoons and look over the site, and then they can appreciate marine biodiversity as much. And so these are what has been happening in Singapore for the past 30 years. From our experience, we find that restoration in Singapore is necessary and relevant, but it's also possible, it's achievable. But what we must do to enhance the success is we need regular monitoring and adaptive management. If things go wrong, we have to step in as fast as possible to make sure that it becomes right again. So we used to think that we want to restore our degraded reefs to improve resilience, but increasingly what we feel is we really have to start thinking about creating new reefs to prepare them for the changing environment. So that's all I have for today. Thanks for listening.
Okay, thank you. It's very clear presentation and insightful information, Lionel.
Thank you.
And now we are opening the question and discussion from the participant. Please, you can raise your hand and also write the question in the chat column. Okay. Maybe during waiting for other question, I also have a question, Lionel, about the propagation of coral reef in the lab. Actually, what is the key point or the key for the successful propagation of coral reef in the lab?
Yeah, are you asking about our work at the St. John's Island National Marine Lab?
Yeah.
So we are testing out different species now. We know that different species, they provide different functions once you transplant them out. So of course, we want, for a restoration project to work, you first need to know what kind of functions you want. Naturally, we cannot put back all the functions that were lost. So it's important to identify the functions that we want. And so for that, we will look at propagating certain corals that have certain functions. So that's what is important to us now. We are looking at the species level. Basically, for example, which corals provide more habitat, more microhabitats, which corals they photosynthesize more, which corals are more thermally resilient. We are not at the stage of heat stressing the corals to get thermally tolerant propagules now, but we are at a stage where we are trying to identify species or genotypes that are more resilient to stress.
You. How about the others, please?
Yeah, I have a question. Very interesting talk, Lionel. It's amazing how like to see the serious approach of the restoration program that you have. But I was wondering is from your talk, it doesn't seems like what scares me is the plan to expand the land in Singapore, and I've seen that starting to happen in Indonesia in many places, although we are not sort of land, but the intention to do reclamation is so huge, maybe because there's a huge money in it, so I was wondering, like, is the government care about the corals? I mean, if they plan about this land expansion, are they doing also, like plan to restore them, or what are they plan to do that? I mean, like from the study from back in January that was published, is obviously like our government has a lot of intention to do a massive restoration in Indonesia. It's just like, techniques need to be addressed properly. But is that the same case with the government in Australia? In Singapore? Thank you.
Yeah, you raise a very good point. In Singapore, we lack space, and that's why government has there's always been a need to increase our land area to support our growth economically and population-wise. According to our very senior profs, the government didn't used to be that keen on this kind of habitat restoration work decades ago, 20, 30 years ago. But increasingly, we do see that they are more open to talks with us, with other stakeholders. They are increasingly starting to recognize the importance of coral reefs. So even though there are still plans, and these are projections toward 2030, and we know that we will still lose some reefs because they still need to reclaim, there are other avenues to still rescue the reefs that might be lost. They are open to relocating corals that might be in the way of destruction to other areas, so we don't completely lose them. So, for example, that video which I showed just now, they were corals from an area in the west of Singapore, which was meant to be a megapod in 2030. So naturally, corals were going to be destroyed. And they discussed, and they were okay with the researchers and environmental consultants shifting as much corals as you can outside and putting them elsewhere where they'll be safer. So definitely, yes, some losses are to be expected, but I think we are optimistic that we can still preserve some of the genotypes and functions. And increasingly, they're also working with different stakeholders. For example, we have a stakeholder group called the Friends of Marine Park, which includes government, researchers, different members of community. Basically, every quarter we sit down, we discuss what are the upcoming plans, how best we can protect our environment, how best we can carry out restoration and conservation work. So I think it's not too bad yet. At least more people are aware. More people in the government are actually more environmentally trained. The younger people, as they move up, the syllabus teaches them that these are important things. So I think they'll be more aware of such things.
Okay, thank you.
Okay, Mbak Ade, it's enough.
Yeah. Like maybe other people would like to ask questions as well.
Yeah, how about the others, please? If you have something to discuss or to ask to Lionel.
Sorry, I have one more. Sorry. So Lionel, do you still continue with this restoration work? Are you still doing it, or have you moved to other projects? Sorry for the background.
It's okay. I'm still doing it. We still have various projects here and there. Of the most recent one is the 10-story high artificial reef thing that I showed you. But other than that, I was also working on my thesis last year about selecting species to enhance restoration outcomes in Singapore. So all these things, as in there's no outright restoration, large-scale restoration program, but all these little projects, all the stakeholders who work together to work on them and make sure that the outcomes are good for general reef conservation and restoration in Singapore.
Okay, thank you. No, the reason my question with that, it's I spoke to Yani before on different subject, but I think it's probably Asian level approach probably would be beneficial,
Yeah.
When we are tackling about the tendency of the monospecific species on a artificial reefs, especially when you expect to have the natural sexual reproduction, and also because I've seen from your photos, you guys actually care about what you place, it's not all the branching, the easy, the fast growing, but also the slow and the plating and everything. And I think that approach need to be also shared with folks in Indonesia, like how actually you're handling this. Yeah, I think that's my opinion. Thank you.
I think it's also because we don't have that many branching corals. So we realized that, or maybe branching corals actually don't work that well in Singapore. It's why we want to try the massives and the platings. And yeah, we do recognize that different growth forms, different species, they have different uses. And that's why we are interested to find out how planting different species gives us different functions. So yeah, I agree that a lot of restoration projects, they focus on branching corals because they give you results very fast, right? Yeah, so I think it's up to us to actually convey this message of less monoculture and more diversity.
That's really true, because when you go to a murky areas, sedimented reefs area, you see less of the branching corals, yeah. And like this is probably useful also to be shared to some of friends from the eastern part of Indonesia that having this problem. Yeah, thank you.
Thank you.
Okay, we have other question in the chat. So with a rising sea surface temperature, is there any evidence if this threat giving pressure to the restored reefs or in natural reefs in Singapore?
So we have had, thank you for the question. We have had 3 major bleaching events so far, 1998, 2010, and 2016. In between, we had minor events in 2013, 2014, and a bit here and there. So we do notice that on the reefs, different species are being affected, even for our restoration projects. So for example, our corals in the tanks or transplanted corals, they do respond differently. So you would expect certain branching corals to bleach and die, but surprisingly, some of them don't. It's the massive corals that end up bleaching. And also for corals that bleach, you might think that they might die, but somehow they suddenly bounce back. So yes, I guess this threat does pressure our natural reefs and restoration projects. But at the same time, we're also using the opportunity to identify what works, what species work, what doesn't. It's unlikely that we'll be able to find out as much as possible before the next bleaching event. But we're just crossing our fingers and trying to note down everything that we can now. But the good news is that at least for the last major bleaching event in 2016, the reefs managed to recover in terms of coral cover in about a year, slightly less than a year. So coral cover did return, but of course, species diversity is a different matter. So we will lose some things, but and gain others, I guess.
Okay, thank you for the question. How about the answer, Pak Veda? It's enough or
Can or not?
yeah? Any others? Lionel, maybe this is out of the topic about restoration, because now I'm working in invasive species. So I have a question. Is there any report or research in the coral reef in Singapore about invasive species? Because as you mentioned before, in Singapore, we have now the shipping activity is very crowded, and then many ship come from other country and maybe can take several invasive, which is trade the coral reef there. So is there any report or information about that?
I think for corals wise, there are no invasive corals in Singapore, but if we're talking about things like the mussels, things that come through the ballast water, then I think there are some instances. But I heard that there are not too, the problem is not too big. I don't really work with invasive species, my colleagues do, so I might be giving the wrong information. But from what I have heard, it doesn't seem to be too big a problem for the marine environment, more for the freshwater environment, where people bring in aquarium species to release once they get tired of them. I think in the marine environment, people do release their pets also once they get tired of them. I am not sure whether it's a major problem. Maybe it's not a major problem because we haven't looked at it on a deeper level. But my sense is that it's not that big of a problem for Singapore yet.
Okay, yeah, because 2 weeks ago I have a discussion also with the ministry from Singapore and also from Malaysia to discuss about same risk area in the waters of 3 countries. That's why I asked. And is there any question? Bang Begin, maybe? Hello, Bang Begin.
Hello, Dea.
Yeah, Bang.
Yes. Yeah, I think this is a great discussion. Yeah. I thank you very much, Lionel, for the sharing based on the Singapore experience. Yeah, I think Indonesia have a lot of challenge, yeah, compared to Singapore.
I think we have different challenges. We just find a way to deal with the challenges in each of our countries.
Yes, but I think a lesson learned from Singapore is very important for us because the closest area, yeah, is Singapore. I don't know in Malaysia. I think compared to Singapore, yeah, they have not so many restoration activity. I think in Malaysia probably. What do you think, Lionel?
I think there might be more, but maybe in terms of documenting these activities, maybe less. In Singapore, it's very small, so everyone knows it, every other person. So that's why we're able to consolidate all this information and present a review, which makes your work, 3 years work in Indonesia, even more fantastic because so many islands, so much work. I don't know how you did it. Yeah, but I think it's important that as a region we work together, find out best practices, what works for each of us, maybe modify what works in each of our respective places, and hope that we get the best results out of this. Which I think there's a potential since we all know each other somehow or other. So it should be easy to talk to.
Yeah. And do you have a recommendation from Malaysia? Is there any colleague that you know that can we invite to share about the restoration in Malaysia?
You know Effendi from Malaysia, I think USM or USM, I think, or Zarina Wahid.
Oh yeah, I know Zarina.
Yeah,
Yeah.
I'm not sure.
Zarina Wahid in Sabah, ya?
Sabah. Zarina is Sabah.
Oh, I met her.
I'm not sure whether she does much restoration work, but maybe Effendi Amri, he may do it. So
Yeah, she worked in biodiversity, I think, probably.
Yeah, so these are possibly people you can ask to share.
Okay, thank you, Lionel.
Thank you.
I hope can visit you.
Yeah. Oh, speaking of which, next year is our Asia Pacific Coral Reef Symposium.
Okay.
In Singapore. So
PCRS.
Yes. Yeah, the last one was in Philippines, right? Not such a long time ago.
Did you go to Philippines?
Yes, I was.
Oh yeah.
I think in Singapore, right? Next one.
Yes, next year. So maybe I'll put in the link here. So hopefully nothing happens in terms of the pandemic, and then we can just meet up and discuss stuff. So I'll put the link here. Yeah, so it's, I think, in June 2023. So looking forward to see all of you.
Reunion, ya.
Yes,
very near also.
I'm sorry seems to ask too many questions, but I have another one that I forgot to ask you, Lionel. Sorry for the back sound. I was like in this big family meeting. My question is like, when you decide where to do the restoration, do you do feasibility studies? And what characters of the reef areas or the water quality that make you choose that site? Thank you.
Yes, last time we didn't used to do as much, but increasingly we're beginning to realize that the site that you transplant the corals to has to have number one, the right environmental conditions, good water flow, and also the other point would be whether it even has corals at all. Because if it doesn't have corals, then there must be a reason why the corals are not surviving there, right? So I think these are the two broad things that we're looking at, whether there's a chance for the corals to survive based on whether there are corals, naturally growing corals there in the first place. And of course, maybe monitor some of the parameters, salinity, temperature, for example. If these more or less match your donor site, then I think there's a good chance for success. And I think you have quite a lot of sites in Indonesia that you can test out all these things, right?
That's true, but how our problem is quite different, especially with the areas damaged by the blast fishing, So that's the tricky one, because the rubble stabilization, it's when I saw your photos, the type of rubbles is quite different. Like, your rubbles is more like degraded because of the sedimentation. But with the blast fishing, it becomes really small rubbles and a large amount of them.
How small?
Like this, very smaller. Yeah, like really small with the blast fishing. And the problem is they keep eroding the healthy corals nearby as well. With your from your just observing from your photos, is only like in Singapore, it's more or less the corals being crumbled, because of the suffocating from the sediments, but here it's like a one-time big blast, and then this damaged area usually take decades. They're still like that, after 30 or 40 years ago. Yeah.
Can I ask what happens to these blast fishing areas? Do you collect the corals back, or because of the local legislation, you are not able to do such things? I'm sure it's very complicated over there.
Yeah, that's a very complicated matters, and especially with the blast fishing happened already a long time ago. So we have the after effects that been decade being, and this area also maybe you've seen a paper before, like where the damaged corals area actually quite becoming toxic, for a healthy corals to settle because of the presence of the algae. It also escalate the challenges.
Okay. Is there any question from others? Yeah, if there is no question, maybe we can close this session and then meet with another session, yeah. Until what session, Mbak? Sorry, you're still mute.
We still have a couple of speakers until the end of next month, but because of the Ramadan coming up, and we still have to discuss that. I can't remember who will give the
20 or 16, Mbak? 16?
Yeah, maybe until the third week of April still have the scores webinars, but maybe the last week of April will be off for 2 weeks, maybe. I don't know, we still have to discuss that.
So this is very nice sharing and full of information, and we hope we can have more activities and collaboration together, yeah, between Indonesia and Singapore, and also join the symposium next year and then meet in Singapore. Okay, Mbak Ade, before we close, is there any information to share with the participant or enough? Bang Begi?
I think enough. Just thank you so much, Lionel, for your time and for your willingness to share.
Thank you very much for inviting me.
I personally quite impressed, with how you put effort into picking up species and do the in situ rearing before replanting to the real reef, So yeah, I hope, like they said, we will continue with the future collaboration and a joint effort. And here's Bang Feda here, who is the WWF CTI for the Coral Triangle and covering the Asian. So this is a big first step to see how we learn from Lionel from Singapore case. I think that's all from me.
And yeah, because I show from the presentation the laboratory in NUS. I think it's good there. If is there any possibility we also maybe can ask or propose to for visitation and also student field trip, yeah, to Singapore, but maybe for discussion.
Yeah, so Trias knows Yani very well. You just had to ask Yani, who is our deputy director for this St. John's National Marine Lab. I'm sure she's more than happy to welcome you guys.
Okay, so it's nice to hear.
Yeah.
Before we close this session, Mbak Cici as a host, please to take a picture for our documentation. Mbak Cici.
Yang ada di tempat bisa nyalain kamera, cuman kita bertiga, Dek.
Ya, okay. It's okay. Okay, ya. Okay, 1, 2, 3. Oke, Bang Cacul, thank you very much for coming. Bang Begin, Iqbal, Bang Riza.
Ya, terima kasih, Lionel.
Thank
you, everyone.
Yeah.
Thank you, Lionel. Thank you, Lionel.
Izin leave. Thank you so much, Lionel. Say hi to Yani.
Okay, see you guys. Hope to see you soon.
Okay, thank you. Izin leave ya. Thank you, Lionel.
Bang Cacul, jangan leave dulu.
Oh ya, oke. Ada